Well, it's my privilege to introduce our next speaker today, Dr. Lindsay Bruce. Some of you may have met me before, but I'm a professor at Knox Theological Seminary. My name is Josh Bruce, and you may notice there's a similarity between our last names. I want to assure you there's no bias in what I'm about to say about our next speaker in terms of an introduction. In fact, to make sure I'm not biased, I actually brought up her official biography so I could make sure to do a good job of this. According to Dr. Lindsay Bruce's official biography, she has a PhD in integrated biology focused on oxidative stress and its effect on neurodegeneration and aging. I practiced saying that 5 times before getting up here. Her research has been published in a number of very distinguished scientific journals, and she has presented on topics as varied as gut microbiome, aging, and best practices for teaching science in the classroom. The one thing that I will say that you won't find on her official biography, and it's the reason I feel most privileged to introduce her today, is that our next speaker, Dr. Bruce, is the very best person I know. And that's because she spends every day changing lives, mine included. So join me in welcoming our next speaker, Dr. Lindsay Bruce.
Well, hi. Thank you for that introduction. I'm Dr. Lindsay Bruce, and I teach biology across the street at Westminster Academy. So I'm going to first have a shout out to all my students who are in the audience today and my fellow science department teachers. My research background is in genomic manipulation of Drosophila, or fruit flies, and I used the transposon-based genetic genomic manipulation system to modify oxidative stress-related enzymes in the fruit fly and see its effects on aging and neurodegeneration. So I love all things genetics, and I'm really excited today to delve into the future of genomic manipulation in humans. So I'd like to start out with a story, one you may actually be familiar with because it made headline news. It sounds like something out of a science fiction novel, and yet it is the world we live in today. On November 25th, 2018, Chinese scientist He Zhengkai stunned the scientific world when he announced on a YouTube video the birth of twin girls that had been genetically modified by his lab as embryos. 2 days later, He presented his research at the Second International Summit on Human Genome Editing in Hong Kong. The twins, named Lulu and Nana, were born to a healthy mother and an HIV-positive father. The gene selected for editing was the CCR5 gene, which codes for a protein that allows HIV to infect cells. The idea was to create children that were immune to acquiring HIV, especially as these particular children had a slightly increased risk of acquiring the disease compared with children from 2 healthy parents. The backlash to this announcement was swift. He's presentation in Hong Kong was crowded with skeptical scientists voicing their concerns. First of all, that it was unnecessary. Why was it necessary to resort to germline modification, which is modification of egg and sperm cells, to prevent acquisition of HIV, when the chance of acquiring the disease was so minimal to begin with, even in the case of the twin girls? Secondly, that it was unethical. Why did He ignore the global community's firm stance on extreme caution prior to human germline editing? He's method was not 100% accurate and could potentially edit other genes besides the targeted gene. He has insisted that neither girl has off-target mutations, but as his work has not been published or peer-reviewed in a scientific journal, it is hard to accurately analyze that claim. Off-target edits created somewhere else in the baby's genomes could lead to cancer or another genetic disorder. Also, that it was impractical. Why did He inactivate the CCR5 gene when the babies could theoretically still acquire HIV through another gene, the CXCR4 gene? While the girls may be less likely to acquire HIV if the procedure worked perfectly, they would not be completely immune. And finally, the unknown. What ramifications would this have for the lives of the baby girls? And what ramifications would it have for any potential future offspring of the twins? Because all of the girls' cells were in theory genetically modified, the modification should in turn pass down to any offspring they may have. He could very well have made healthy babies sick and caused their offspring to have the disease as well. The work was quickly denounced by the conference organizing committee, followed by condemnations from the US, the UK, Chinese, and other governmental leading scientific organizations. Leading scientists such as Jennifer Doudna, the co-discoverer of the CRISPR-Cas9 genome editing tool, which was used to modify the twins' genomes, also publicized their concerns. And then the situation got even crazier. He returned to China and disappeared, as in no one knew where he was. The watching world was left with many questions. What had happened to He Zhengkai? And what about the health and well-being of the world's first ever germline edited humans? So let's take a step back from this saga for a moment and ask a question. What is gene editing anyway? To answer that question, we must start with our genomes. Our genomes consist of all the DNA in our cells. You probably recognize DNA in this picture in all its double helix glory. DNA gives instructions for making proteins, which carry out a huge number of functions in our bodies. Proteins act as structures, like the actin and myosin filaments seen here that make up our muscles. Proteins act as transport molecules, like hemoglobin, which carries oxygen around to our bodies and our red blood cells. Proteins act as enzymes, like the enzyme catalase, which breaks down harmful hydrogen peroxide in our body into non-harmful water and oxygen. You've actually probably seen catalase at work before and might not have even known. If you've ever had a cut and washed it out with hydrogen peroxide, you'll have noticed that it bubbles. That bubbling is the result of the catalase in your body decomposing the hydrogen peroxide that you poured on it and releasing oxygen. Your DNA is packaged into chromosomes, and you get half your chromosomes from your dad, pictured here in blue, and half your chromosomes from your mom, pictured here in pink. And the process of handing down chromosomes from parent to offspring works very well. However, genetic diseases like cystic fibrosis, spinal muscular atrophy, and sickle cell anemia can be passed from parents who are carriers of a disease to the offspring. In other cases, random mutations, changes to the DNA code, can occur and cause a genetic disorder when neither parent was a carrier. While there are treatment options for genetic diseases, there are no cures. Scientists have been searching for a cure to genomic diseases through genome modification for some time now. Initially, scientists used gene therapy. Gene therapy uses an inactivated virus, like the common cold virus, to deliver a healthy copy of a gene to a patient with a mutated copy. It doesn't correct the mutation, it simply provides a good copy alongside the mutated copy. In the blue diagram here, you can see the viral vector, in this case the adenovirus, and it contains a new gene, shown in pink, that has been modified and placed inside the viral vector. The viral vector then directs the gene to the nucleus of the cell, where the other DNA is, and the protein-making machinery is so that the cell can take the DNA and turn it into a new protein to help the cell. The first time that gene therapy was used was in 1990 in a 4-year-old patient, a 4-year-old girl who had adenosine deaminase, a disease that left her without an immune system. And so this therapy was used in this patient very successfully and in other patients following her, and she was able to be, at least some of her symptoms were alleviated from this particular disease. However, in 1999, 18-year-old patient Jessie Gelsinger, pictured here, volunteered for an ornithine transcarbamylase, OTC deficiency, gene therapy trial. Jessie had the mild version of this disease, but it was controlled through diet and medication. He volunteered for this trial, trying to help the patients, the babies who were born with a very severe version of this disease and would die in infancy. However, Jesse had a massive reaction to the viral vector that was used and died. Immediately, all such gene therapy trials halted. Gene editing is slightly different than gene therapy. Instead of simply giving good copies of a gene to a patient with mutated copies of a gene, gene editing has the potential to repair the mutation within the person's genome. The first types of gene editing molecules that were discovered are known as zinc finger nucleases. They're pictured here under the letter A. They were first discovered in 1985 and first used to edit the genome of mice in a mouse cancer therapy trial in 1994. But zinc finger nucleases were expensive, time-consuming to make, and had a very low efficiency. That is, they didn't work all that well. The next gene editing complex to be discovered, transcription activator-like effector-based nucleases, or TALENs, is pictured here under the letter B. TALENs were found in 2007 and first put to use editing the genome of crickets in 2012. But while the efficiency of TALENs was much higher compared to the efficiency of the zinc-fingered nucleases, they were still time-consuming and expensive to create. And they were quickly overshadowed by our last genome editing complex, clustered regularly interspaced short palindromic repeats, or CRISPR, pictured here under the letter C, and a story about cheese. Yes, I meant actual cheese, and my apologies for mentioning cheese this close to lunch, but please bear with me. Dr. Sylvain Moineau and his lab at Larval University in Quebec was performing research on the cheese-making bacteria Streptococcus thermophilus. The problem was that this bacterial strain kept getting infected with a virus. Those yellow spider-like creatures you see on the bottom right corner picture, those are bacterial viruses. Fun fact, bacteria can get sick with viruses just like you and I can get sick with viruses. And when your cheese bacteria gets sick with a virus and dies, the milk never becomes cheese and the world mourns. But just like humans, bacteria also have immune systems that allow them to fight off certain viruses. And Moineau and his colleagues noticed that some bacteria were better off at fighting the viruses versus other bacteria. And this was because of the bacterial's defense system, which we now know to be the famous CRISPR complex. Other scientists working for DuPont's food production company, Danisco, continued to build upon this discovery. So Rodolphe Barango and Philippe Horvath discovered that CRISPR worked by actually keeping a piece of the viral DNA in its own genome, kind of like a red flag and almost like a vaccine works. It uses it to identify if it gets reinfected with the virus. I've already seen this virus and I need to destroy it, and sends out the CRISPR machinery to immediately destroy the virus. Now, it's quite a long way from bacterial defense system to curing human genomic diseases. And that's where two researchers, Emmanuel Charpentier, who's now at the Max Planck Institute in Berlin, and Jennifer Doudna of the University of California, Berkeley, come into the story. Charpentier showed how RNA molecules lead CRISPR to a specific section of DNA. And then Charpentier and Doudna collaborated to show how Cas9, which is CRISPR-associated protein number 9, cuts the DNA at a particular section, and how this could be used to actually modify DNA in a living organism. George Church of Harvard and Feng Zhang of the Broad Institute, MIT, showed that the system could be successfully used in human cells as well. This diagram shows an overview of how the system could repair DNA. The Cas9 protein is seen here as a white cloud, and the guide RNA is seen in purple. They form a complex. The guide RNA then takes the Cas9 to a specific segment of DNA, where Cas9 then cuts the DNA completely through. At that point in time, the DNA is available to be programmed, and you can put in a pre-programmed or a newly modified piece of DNA into the DNA at that section. So CRISPR-Cas9 research spread like wildfire. In 2012, when Charpentier and Doudna published their findings, there were only 126 papers published that entire year from all researchers on CRISPR. But last year, there were over 5,000. Clinical trials using CRISPR-Cas9 are gradually starting to pop up. The most encouraging of these types of trials are treating blood diseases, like sickle cell anemia or beta thalassemia. In the sickle cell anemia trial, which is pictured here, the only patient who has been treated so far is Victoria Gray, who is a mom of 4 and has been suffering debilitating pain attacks since she was young. Sickle cell anemia causes blood cells, which should be round, as seen in the picture to the right, to become sickle-shaped. The sickle-shaped blood cells don't move well through blood vessels and get caught in blood vessels and in joints, and this leads to pain, swelling, inflammation, and other symptoms. Last year, Dr. Haider Frangel of the Sarah Cannon Research Institute in Tennessee removed Miss Gray's bone marrow, corrected the deficiency in her blood cells using CRISPR-Cas9, and then reinserted those blood cells into her bone marrow again. Less than a year later, her blood is making fetal hemoglobin, the protein that she was once lacking. And incredibly, Miss Gray has not suffered a debilitating pain attack since her procedure. Researchers at the University of Pennsylvania are using CRISPR to fight off cancer using a type of therapy called CAR T-cell therapy. Blood is drawn from the patients, and T cells in the blood are weaponized into CAR T cells that target and fight cancer cells. It's your own cells that are going to fight the cancer for you. These cells are then multiplied and then re-injected into the patient. Now, this type of treatment has been used before, prior to CRISPR, but in those clinical trials, the problem with it was the patients had a pretty good immune response to the modified CAR T cells. In this particular trial, in multiple myeloma and sarcoma patients, the patients exhibited no side effects to the CRISPR-edited CAR T cells. However, the dosage is not quite right yet, as it did not actually effectively treat the cancer. So further clinical trials are going to continue to allow it to halt and modify the progression of a specific cancer. There are quite a few biotech companies that are plunging forward with CRISPR-based advances. Editas Medicine is developing therapies for various genetic blindness diseases, such as Usher syndrome and Leber congenital amaurosis, or LCA. They have one clinical trial underway in LCA that involves both gene therapy, the one we discussed before, and gene editing. They use the viral vector to transport the CRISPR-Cas9 machinery to the correct cells in a person's eyes, where the CRISPR-Cas9 machinery can correct those cells and restore sight. Now, these types of trials are less problematic than having a gene therapy throughout your entire body, because your eye cells have very little immune response. And so the likelihood of getting some sort of crazy reaction to this type of gene therapy is minimal. Another biotech company is CRISPR Therapeutics. This one was started by Charpentier, the researcher that we discussed earlier, and it's looking at immune therapies for cancers, as well as they have a small beta thalassemia, a blood disorder trial running in Germany. Mammoth Bioscientists was started by Jennifer Doudna, and she's using CRISPR-based products to allow doctors to rapidly diagnose genetic diseases, which previously had taken more time. Because this technology is inexpensive and accessible, there has been a whole new movement of do-it-yourself, DIY bio, that has exploded. These movements, as a scientist, I think are kind of neat. The goal is to open a lab space to the public. Then anyone can come in and take a class or use the tools and supplies that are available there to play around and do their own research. They use them to do inventions of many different kinds, to take things apart, to rebuild things. They use them even to create bioart, which is really interesting. This particular lab is called GenSpace, and it's in New York and led by a biologist, Ellen Jorgensen. But researchers, biotech companies, and DIY bio-open lab spaces are not the only people using CRISPR-Cas9. A whole new branch of scientific researchers has popped up using YouTube and home labs in their garages. The unofficial leader of this movement is Dr. Josiah Zayner, pictured here. He's a former NASA employee and a self-proclaimed biohacker, and owner of the biotech company Odin, which has the goal of making CRISPR-based technology accessible to the masses. Zayner conducted a CRISPR experiment on his own muscle in front of a live audience in 2017. He was also drinking scotch from a flask while he did it, so it was a crazy experiment. You can view it online. The injection was meant to increase the size of the muscle cells that came in contact with the injection, but it seems to have only resulted in some amount of redness and swelling. Zayner admits he did not expect it to work, but he did want to do it as a bit of a publicity stunt to draw attention to the technology. One of Zayner's acolytes, Mississippi dog breeder David Ishii, has bought supplies from Zayner and eventually created this lab in his backyard with the hopes of both creating dogs that can glow in the dark using jellyfish protein, and also curing Dalmatians of genetic diseases that have plagued them due to inbreeding. While the US government is starting to put restrictions in place on these types of procedures and practices, the supplies are readily available, and people are taking advantage of it, trained researcher or not. Another way that gene therapy and editing is being used is as biotourism. So Liz Parrish has announced that she would like to live forever. So she traveled to Latin America to have an undisclosed company provide her with anti-aging gene therapy. She has also not disclosed the nature of the gene therapy injections, but has hinted that they involve the enzyme telomerase, which is involved in lengthening the ends of your chromosomes, which eventually shorten as we age. The problem, of course, with this being that it's a very risky endeavor, as cancer cells also are able to lengthen their own telomeres at the end of their chromosomes. She has started her own biotech company devoted to anti-aging called BioViva. Brian Hanley is a professor at Auburn University, and he runs a company that wants tourists to come to him, to the US, for gene therapy treatments, anti-aging gene therapy treatments. He managed to get his own treatment here in the US, and he did this by getting a local ethics board to sign off on the treatments and enlisting the help of a plastic surgeon to give him the gene therapy injections. It's likely that other people are also trying these treatments too, just behind closed doors to attract less scrutiny. And while currently risky, the technology is there to create designer children. We are on the verge of wealthy parents paying for the child they want to have. If Felicity Huffman and Lori Loughlin are willing to pay vast sums for an SAT score, how much would they pay for a genetically engineered child who is able to take the SAT and score a perfect score on their own? All this brings us back to Lulu and Nana, the genetically modified Chinese baby girls, and the scientist behind it, He Zhengkai. After nearly a year of seclusion, it turns out that he was being kept under house arrest by the Chinese government. He is pictured here on the balcony of his apartment during his house arrest. He was sentenced to 3 years of prison time and a close to $500,000 fine. But the punishment feels almost mandated by the public worldwide outcry. China has no laws against human gene editing, and despite the messiness of this situation, does not appear to be moving towards any restrictive legislation on this front. There has been no news on Lulu and Nana, the now 1-year-old girls who were genetically manipulated. Some fantastical reports have indicated that they have above average intelligence, and others have indicated that they may have a shortened lifespan due to the modification. But both reports have been decried by the majority of scientists as premature. Gene editing may, in fact, have no effect on the girls. And given the lack of credible news on them, we may actually never know if it does. Like it or not, gene editing is here to stay. CRISPR-Cas9 has paved the way for a whole new realm of technological advances on that front. And the experimentation continues onward, regulated, unregulated, constrained by ethical guidelines, or ignored by government officials unless it causes too much of a stir, in pristine laboratories, or in your own garage. Thank you.
Thank you, Dr. Bruce, for that very informative presentation. It's kind of a hard act to follow. Let me just say at the outset that in many ways I am the worst person in the whole entire world to give a response on science. And to just explain this to you a little bit, let me tell you what I did when they asked me to give this response. I Googled the word gene. Now, Google initially thought that I was looking for the proper name, Gene, short for Eugene. So it told me that the name is of English origin and means well-born, but that didn't help at all. So I was a little stuck. For those of you who are listening to all of this and maybe find yourselves a little confused at points, you're in good company. You're in my company, so company. As a few of you will know, perhaps, I teach at Knox, and my background is in church history and in classics rather than in science. So, like many of you, I am not going to have a PhD in science, and I have almost no background in science whatsoever. Science scared me as a kid for reasons that were brought home as I sat here and listened to this. It sounded like a brave new world, and it kind of still does. I suspect that many of us here today are asking certain questions as we hear this kind of presentation when it comes to the editing of genes. Most of the time, I think, when people like you or me are hearing these kinds of talks, we're not so much asking technical questions, although some of the technical details may be a bit over our head, but I think we're usually asking more spiritual questions, moral questions. So we're asking things like, not how do we do this, but should we do this? Is it moral? Is this consistent with what we believe, and many of us here are Christians, about what it means to be human? What it means to be created in the image of God? So Dr. Bruce, on behalf of our audience, if you don't mind, I wanted to ask you a few questions here about all of this that come from the perspective of someone like me who's in a ministry context rather than a science lab, or it turns out a neighborhood garage, I guess, if you're doing gene editing. Hopefully, these questions can at least begin a very important conversation that I think we should have about a very important issue. I would encourage you, as Dr. Sansbury encouraged you, to write down questions as well of your own, because you will have a chance later on today to ask those. So if it's okay with Dr. Bruce, I'll go ahead and ask 3 questions. I should mention to you all that these 3 questions are written down, so I'm going to read them, but Dr. Bruce has not seen them at this point. That was not strategic. I used to be a trial attorney. I'm not pulling a fast one. Dr. Bruce and I are married, and we have 2 little children, so what that means is we don't talk. Until today, actually, so thank you all for this opportunity. We get a chance to chat. We should do this again. Here are the 3 main theological and pastoral questions that came to mind as I reviewed Dr. Bruce's talk. The first one is one that I suspect many of us were kind of wondering as we sat here today, and it's this question: are we playing God? Isn't the sort of research that Dr. Bruce has told us about flowing out of a sinful kind of pride? The arrogance that assumes that we can know as much as God, and actually maybe we can know better than He does. Shouldn't a Christian response to this sort of technology be a humble acceptance of our place in God's good and created order? And I suspect that at least some of you were wondering that as well. Peter Bruegel's magnificent Tower of Babel came to mind as I wrote this down, and you'll see it up on the screen there. And I sometimes wonder if our genetic scientists these days aren't just modern versions of Nimrod and company scaling heaven to overthrow God. The fact that they wear lab coats probably causes us more concern rather than less. I mentioned earlier that I have some background in classics, and I'll tell you from that background that you don't have to be a Christian to share some of these concerns. Even going all the way back to the ancient Greeks and the ancient Romans, they all had stories about what could happen if we exceeded the bounds of our human knowledge. They have warnings about the dangers of pride. Think of the stories of Pandora's box or the story of Icarus. If you know those stories, you know what I'm talking about. The penalties that we pay for flying too close to the sun. So here's my question to Dr. Bruce. Do scientists in this field share that kind of concern at all? And I'll make one caveat as I ask you that question. Please do not tell me in your answer, I beg you, that if I like taking aspirin for a headache or prefer Novocaine when I'm having my teeth drilled, that I should also support genetic engineering. They don't feel the same to me. My second concern is focused on the equities, or perhaps I should say the inequities of all of this. As Christians, and especially as Christian leaders, as many of you will be in different ministries, we all are called by Scripture to care for the powerless, the poor, the widow, the orphan. The Bible just doesn't leave us any choice on that. But let's be honest and let's admit it. If scientists are being honest to us, wouldn't they have to tell us that, especially with respect to the modification of embryos, the people who are going to be able to afford this kind of treatment are going to be first and foremost the richest among us, those who can afford to pay large sums. The SAT cheating scandal that you mentioned, Dr. Bruce, is just one recent example of the lengths that so many wealthy parents will go to secure the very best opportunities for their own children, even at the expense of so many others. Personally, and I hate to be too dramatic about it, but I dread a future dystopian Lake Wobegon. Y'all know Lake Wobegon? Where all the rich women are strong, all the rich men good-looking, and all their rich little brats above average. I fear it. Now, Dr. Bruce, you and I have two fine little geniuses of our own who I'm sure will hold their own against their genetically enhanced peers, but I fear for our grandchildren. How are they going to get into Harvard if they're competing against 7-foot-tall NBA athletes who are also born with a genetic predisposition to understand Hegel? I have to say, even as I'm saying it, it does sound pretty cool. More seriously, though, does a future in which our wealthy parents shop for blue-eyed, blonde-haired, Nordic athletic geniuses bother you? Does it bother other scientists? I've been told, of course, that the minute you bring the Nazis into an argument, you've lost it. I've just done that, and I'm not even a little ashamed of it. It's what we're all thinking, right? Lastly, and this is a bit more of an open-ended question than my first two questions, what, if anything, Dr. Bruce, keeps genetic scientists up at night worrying about all of this? I suspect that there might be some real concerns that genetic scientists actually feel, but perhaps they're worried about sharing them with the rest of us for fear of panicking us. So I ask you, as I conclude these questions, to just give us maybe one or two of these sincere concerns that genetic scientists might actually have. And I say that so that we can be better informed, but also so that we can all get very upset stomachs before our lunch. Thank you.
All right. Well, I'll do my best to take a shot at some of these questions. I also apparently need to remind the dentist never to give my husband Novocaine. Okay. So I think the first question that I heard was, are scientists playing God? I think that's a bit strong of a way of putting it. I think the fear that is being expressed in regards to this technology is kind of the same fear that has been expressed by almost every age in response to a new advance or a new technology. And the simple fact is this, that like it or not, this technology is being implemented now, full stop. So it's hard to say that we should completely stop this because at least in some areas or unregulated scientists or unregulated areas of the world, that this is going to continue. And even in the US, versions of this are going onward. So I think that we have to deal with the technology and have the conversations about the technology that we have that is being implemented now instead of having a kind of a fear-mongering type thing where this is scary and this could go wrong. With that said, I think that there are a lot of concerns that should be voiced in response to this, and I think they are being voiced by the scientific community, by bioethics communities. So I think that there is a lot of pushback on implementing this carefully and avoiding just willy-nilly trying to make ourselves some best version of ourselves that we could be. So that was the first point I heard. The second point I heard was about the inequality of it and the creation of designer babies. So I think there are kind of two categories of implementation of this, at least with regards to enhancing ourselves. I think there's kind of this level playing field where kind of a healthy person is, and most people, I think, don't have a problem with trying to carefully and cautiously move forward on helping those who have a genetic disease kind of come up to where everyone else is. So curing or alleviating suffering is generally seen as a positive use of this technology. The question is, if we take a normal human and then try to make them superhuman in some way, is that ethical, and should it be ethical, and how should that be regulated? The biohackers that we see are trying to do this to themselves, and I think the government is starting to try and regulate that a little bit, but it's often been seen as, if you want to try this on yourself, go ahead, but that we definitely need to have some serious conversations before anything like that. Taking a normal person and trying to enhance them is move forward. I think there's a lot more caution that needs to be taken with that particular use of the technology. And I think the immediate inequality isn't going to be designer baby versus regular baby. I think the immediate inequality may be affordability and access to treatment for patients with diseases, because even now, CRISPR or non-CRISPR based medicines are sometimes so expensive that lower to middle income people don't have access to the treatment, the ability to treat their babies. So it's kind of, I think as Christians, since we care about humans made in the image of God, that one of the things that we need to be thinking about is how to equalize access to treatment, so that it's not those who can afford to save their children's lives through this new groundbreaking treatment versus those who cannot afford to do this. And then lastly, scientific concerns. Does the scientific community have concerns? They definitely are voicing concerns. There's been, scientists, including Jennifer Doudna, who I mentioned, have gone in front of the US government on panels and talked to ethics committees. And there's a whole branch of bioethics that is trying to allow scientific communication, scientific dialogue, so that it's not just ramrodding forward implementation of this without people knowing what's going on, but allowing people to actually see, okay, these are the possible uses. How do we want to use this? How do we want to move forward? There are, crazy ways in which this technology could be used that are concerning, that are scary. So I can think about like bioterrorism or bioweapons if you wanted to modify the bubonic plague virus, or bacteria, not virus, to be, somehow unable to stop by the current methods that we have, and use that as an agent of biowarfare. That's like a crazy way in which this could be taken. I think the concerns on that front may be slightly overblown, just because if you have, there's so many, unfortunately, weapons that are available for destruction. That seems like a very long way around to get to the same point that other things can help you do. There could be enthusiastic biohackers where it goes wrong, and so you have problems with that. There was a biohacker last year that was found dead. They don't think it was related to his biohacking, but the CEO of Ascendance Biomedical was trying out all kinds of crazy things, but was probably not biohacking related, but he was found dead probably due to ketamine, I think. And I think scientists are mainly worried about germline edits, so edits to sperm and egg cells, because those are the edits that not only affect the person being that will be edited, but the future generations. And how that is passed down through the generations, I think, is something that we don't know yet. And it'll be, eventually, I guess we'll see how that goes. But right now, the technology is risky to do that at this point in time. So that's about all I can think of.
I think one of the really interesting things that Lindsay talked about in this is it's great to have the conversation about what should be done, but it's not just a conversation about what should be done. It is going to happen, and people are experimenting on themselves, and there's going to be very interesting questions for us in the church pastorally. What do we do when someone walks into the door who has been willfully attempting the effort of transhumanism, of becoming more than human, of becoming something other than human? What will we say and what will we do? And how do we even think that through theologically when one of the main traditions of the church with respect to what Christ did is taking on humanity? What happens when someone has willfully chosen to try not to be a human? So there are some really interesting questions that are going to come, and we don't know how it will go. We don't know what the shape will be. When I was in seminary, we were supposed to have clones walking into our churches all the time by now. It hasn't happened, but it is still a strange world. It was also supposed to be very hard. I remember gene editing, and the statements were, it's very hard, it's very expensive, and we haven't been able to do it well. And now you can buy a kit online.